The concept of human cloning has long been a subject of fascination, controversy, and ethical debate. From science fiction stories to real-world scientific advancements, the idea of creating a genetically identical copy of a human being raises profound questions about identity, morality, and the limits of science. But what is the truth about human cloning? Is it possible, and if so, what are the implications? To answer these questions, we must explore the science behind cloning, the challenges it presents, and the ethical considerations that surround it. Cloning, in its simplest form, is the process of creating a genetically identical copy of an organism. This can be achieved through various methods, the most well-known of which is somatic cell nuclear transfer, SCNT. This technique involves taking the nucleus of a somatic cell, any cell in the body that is not a sperm or egg cell, and transferring it into an egg cell that has had its nucleus removed. The egg cell, now containing the genetic material of the somatic cell, is then stimulated to divide and develop into an embryo. If successful, the embryo can be implanted into a surrogate mother and develop into a cloned organism. This method was famously used to create Dolly the sheep in 1996, the first mammal to be cloned from an adult somatic cell. While the cloning of animals like Dolly has been achieved, the cloning of humans presents a far more complex and controversial challenge. Scientifically, human cloning is theoretically possible, as humans share many biological similarities with other mammals. However, the technical and ethical hurdles are significant. One of the primary challenges is the low success rate of cloning. Even in animals, the process is highly inefficient, with many attempts failing to produce a viable embryo. In the case of Dolly, it took 277 attempts to achieve a single successful clone. For humans, the process would likely be even more difficult, as our biology is more complex and less understood than that of sheep or other commonly cloned animals. Another major challenge is the risk of genetic abnormalities and health issues in cloned organisms. Cloned animals often suffer from a range of health problems, including premature aging, organ failure, and immune system deficiencies. These issues are thought to arise from errors in the reprogramming of the somatic cell's DNA during the cloning process. In humans, such abnormalities could have devastating consequences, raising serious ethical concerns about the welfare of cloned individuals. The potential for harm to both the clone and the surrogate mother makes human cloning a highly risky endeavor. Beyond the technical challenges, human cloning raises profound ethical and moral questions. One of the most contentious issues is the concept of identity and individuality. A clone would be genetically identical to the person from whom they were cloned, but they would still be a unique individual with their own thoughts, feelings, and experiences. This raises questions about the rights and autonomy of the clone, as well as the expectations and responsibilities of the person who chose to create them. Would a clone be seen as a mere copy, or as a person in their own right? How would society treat someone who was created through cloning? Another ethical concern is the potential for exploitation and abuse. Human cloning could be used for unethical purposes, such as creating clones for organ harvesting or as a means of achieving immortality. The idea of designer babies or clones created for specific traits or purposes raises concerns about eugenics and the commodification of human life. These possibilities highlight the need for strict regulations and oversight to prevent the misuse of cloning technology. The ethical implications of human cloning have led to widespread opposition from religious, cultural, and scientific communities. Many argue that cloning undermines the sanctity of human life and interferes with the natural process of reproduction. Others worry about the psychological impact on cloned individuals, who may struggle with issues of identity and self-worth. These concerns have led to calls for a global ban on human cloning, with many countries enacting laws to prohibit the practice. Despite the challenges and controversies, some scientists and researchers continue to explore the potential of human cloning, particularly in the field of therapeutic cloning. Therapeutic cloning involves creating cloned embryos for the purpose of harvesting stem cells, which can be used to treat a wide range of diseases and conditions. Unlike reproductive cloning, which aims to create a living organism, therapeutic cloning is focused on medical research and treatment. Proponents argue that this approach could revolutionize medicine by providing personalized treatments and cures for diseases such as Parkinson's, Alzheimer's, and diabetes. However, even therapeutic cloning is not without its ethical dilemmas, as it involves the creation and destruction of human embryos. In addition to therapeutic cloning, advances in genetic engineering and biotechnology have opened up new possibilities for creating genetically identical organisms. Techniques such as CRISPR-Cas9 allow scientists to edit genes with unprecedented precision, raising the possibility of creating designer clones with specific traits or characteristics. While this technology holds great promise for medical research and treatment, it also raises concerns about the potential for misuse and the ethical implications of altering the human genome. The debate over human cloning is further complicated by the lack of international consensus on the issue. While many countries have banned reproductive cloning, others have more permissive laws or no regulations at all. This patchwork of laws creates a risk of cloning tourism, where individuals or organizations seek out countries with lax regulations to pursue cloning experiments. The lack of a unified global approach to cloning highlights the need for international cooperation and dialogue to address the ethical, legal, and scientific challenges posed by this technology. In conclusion, the truth about human cloning is that it is both scientifically possible and ethically fraught. While the cloning of animals has demonstrated the technical feasibility of the process, the challenges and risks associated with human cloning are significant. The low success rate, potential for genetic abnormalities, and profound ethical concerns make human cloning a highly controversial and complex issue. Advances in therapeutic cloning and genetic engineering offer potential benefits for medical research and treatment, but they also raise new ethical dilemmas and questions about the future of human reproduction. As science continues to push the boundaries of what is possible, it is essential that we approach the issue of human cloning with caution, responsibility, and a commitment to upholding the dignity and rights of all individuals. The debate over human cloning is far from over, and it will require ongoing dialogue and collaboration to navigate the complex intersection of science, ethics, and humanity.